Hi, welcome for formative assessment 5.2, reviewing the main ideas. Draw what a chromosome looks like during metaphase. Identify the chromatids and the centromere. Draw what chromosomes look like during metaphase. So if we are talking about metaphase, then we can have something like so we are having something like this. Then inside it, of course, we are having here the centrioles. Then we are talking of metaphase. So in a metaphase, they are normally aligned at the center. So we have things like this. You have things like this aligned here. So they are normally 4. Let's try to make them look different. So we have things like this, and as I have said, they are aligned at the center. They are aligned at the center. This point here is what we are calling the centromere. Centromere. Then chromatids. Actually, this is chromatid. Chromatid. So this is a chromatid here. As it runs down, then we have spindle fibers. So we'll have spindle fibers running down. Spindles. Spindles. We say the spindles will run through the cell cytoskeleton, so they will look something like this. So we are having chromosomes. These are chromosomes aligned during interphase, during metaphase. So let's see if we are getting right. We can go back to our picture here, and this is how they are aligned during metaphase. They are aligned across. They are aligned across. So we can also align ours across to be exactly make it exact the way they are. So we have something like this. That is how they are actually aligned. So that is what the question expects you to draw, and then you show what is the centromere. Sorry. So it's not the centromere, this is the chromatid. So if this is what we are having here, then this is the centromere, the middle part. This is the chromatid. So let's go to the next question. The next question asks, briefly explain why the daughter cells resulting from mitosis are genetically identical to each other and to the original cell. You should explain to us briefly why. That is what the question wants. The question wants you to explain why the daughter cells here are actually identical. So mitosis is the process that eukaryotes use to make exact copies. Mitosis is a process that the cells use to make exact copies. To make exact copies of their cells. So they actually make exact copies of their cells for growth. This would be for growth, repair, because repair cells have to be the same kind of cells, or asexual reproduction. So even in the asexual reproduction, the cells are exact copies. I mean, the daughter cells should be exact copies of the parent cell. So mitosis divides the nucleus into genetically identical nuclei. So each with its own full set of DNA. Each with its own full set of DNA. Mitosis divides a cell's nucleus into two generically identical nuclei. So it is producing two generically identical nuclei each with its own full set of DNA. Each with its own full set of DNA. So because each of them will have its own full set of DNA, then this will result in genetically identical individuals. This will result in genetically identical individuals. So, and before this happens, or before cell division, all the other organelles are also duplicated to form a copy of each of them before the cell actually begins to divide. So let's go to the next question. Question number 3. Critical thinking. Contrast how do prophase and telophase differ. Contrast how do prophase and telophase differ. So how are they different? So let's try and make a table here and see how we can do this. So if we have a table like this, one side of the table we have the prophase, and the second one we have telophase. So here we have prophase, and here we have telophase. So what happens during prophase? The DNA and proteins condense into tightly coiled chromosomes. Here, the DNA and proteins condense into tightly coiled chromosomes into tightly coiled chromosomes. So we are having here tightly coiled chromosomes. In telophase, chromosomes begin to uncoil. Chromosomes begin to uncoil. So when here they are condensing into tightly coiled chromosomes, in telophase, the chromosomes begins to uncoil. To during prophase, The nuclear envelope breaks down. The nuclear envelope breaks down. What about in telophase? During telophase, the nuclear membrane starts to form. The nuclear membrane starts to form. So here the membrane starts to form. The other one, the membrane actually is dismantling. Centrioles begins to move to opposite poles. Centrioles begin to move to opposite poles during prophase. Centrioles begin to move to opposite poles. And spindle fibers form. And spindle fibers form. On the other hand, in telophase, the spindle fibers fall apart. The spindle fibers fall apart. The spindle fibers fall apart. So if you are making comparison of the two, you can see how they look like opposite of one another. In one, the DNA is condensing, in the other, it is uncoiling. In the other one, the nuclear envelope breaks down, and in the other one, it starts to form. Also, prophase is the first stage of mitosis, and telophase is actually we can call it the last stage of mitosis. So this is first stage, this is last stage. That one can also be a difference. If we go back to the picture, we can see what we mean by these differences. So in the next video, we will try to look at the remaining questions. So we'll stop here for now.